Picture an early human finding a bright, strange mushroom in the woods. They are starving, but eating it is a massive gamble. It could be dinner, it might be medicine, or it could be lethal. Today, we face a different kind of gamble. If you look at how we talk about health, there is a sharp divide. On one side, people who swear by the ancient wisdom of natural, plant-based remedies. On the other, those who refuse to touch anything that has not cleared rigorous FDA clinical trials. We are not here to decide which camp is right. Instead, we are going to look at how these two approaches are directly connected and how traditional herbalism built the exact foundation that modern science relies on. This flowchart shows the fork in the road we hit when evaluating a newly discovered plant. You can take the left path, relying on ancient observation like human and animal trials, or the right path, using the strict protocols of modern science, like microscopes and clinical data. Figuring out what heals us, what harms us, and what alters our minds is an unbroken chain. It is the longest and most dangerous ongoing experiment in human history. To start gathering data, early humans had to be incredibly observant. They watched which berries deer ate and which leaves bears avoided. But ultimately, someone had to test these plants themselves. That testing came with a brutal cost. Identifying which plants reduced a fever and which ones stopped your heart required generations of trial and error, paid for with countless unrecorded human casualties. Because survival was on the line, early researchers documented their findings. Cave walls became the first medical databases, recording animals connected to these discoveries. Over time, these painted records evolved from simple observations into vital communal safety guidelines, warning tribes about toxic vegetation. If we plot this out on a comparison matrix, the empirical tradition for data gathering gives us broad, anecdotal evidence about what works, but acquiring that data carried an incredibly high risk to the individuals testing it. This ancient method successfully identified the correct healing plants, but it required high-stakes beta testing to do so. As tribes grew, they could not afford to have everyone taking those risks. This knowledge concentrated into the hands of specialized healers and shamans who acted as the first living repositories of medicinal data. To prevent highly localized botanical knowledge from disappearing when a healer died, early civilizations upgraded from oral traditions to written manuscripts, formally codifying their remedies. They also tried to create reliable, standardized medical instruments to complement their herbal remedies, showing an early drive for physical consistency. However, they hit a physical limit when it came to dosing. You can prescribe 2 willow bark leaves for a headache, but the actual potency of those leaves changes drastically depending on the season, the soil, and the rainfall. Updating our matrix, we see the physical limits of ancient refinement. The knowledge storage remained vulnerable, and natural variations meant creating a perfectly consistent dose was nearly impossible. Ancient traditions established an excellent baseline for community safety. They simply lacked the technological capacity to replicate a specific dose perfectly every single time. A major shift happened when early modern scientists stopped boiling whole leaves and started isolating the exact active chemical compounds responsible for a plant's effects. Early scientific instruments allowed researchers to push past what the naked eye could see. They could finally identify the microscopic biological triggers causing a reaction. The benefit of this chemical isolation is extreme precision. You can extract the exact molecule you need and administer a perfectly measured milligram dose, targeting specific physiological responses. But there is a scientific trade-off, known as the entourage effect. Isolating one single compound strips away complementary, synergistic chemicals found in the whole plant, compounds that might naturally buffer negative side effects. Looking at the third row of our matrix, the scientific method offers exact milligram precision. The cost of that precision is the loss of the plant's natural botanical synergy. Modern chemistry traded the holistic complexity of the ancient forest for the absolute, controllable precision of the laboratory. To prove that an isolated compound actually works, the scientific method subjects ancient botanical claims to highly controlled stress tests. This separates true physical efficacy from the placebo effect. From there, modern medicine established strict regulatory protocols to protect mass populations from adverse side effects. The trade-off is speed and cost. Running these double-blind clinical trials requires massive financial investment. severely slowing our access to novel treatments. But clearing those intense regulatory hurdles comes with a major reward. We gain the ability to safely mass-produce and distribute standardized medicine globally. Completing our matrix, the scientific method provides universal, evidence-based safety, but it is highly resource-intensive to execute. Modern clinical verification is incredibly slow and expensive, yet it creates a universal safety net that ancient trial and error could never guarantee. If we look at how this data is built, the cultural war between science and nature breaks down. Modern research does not overwrite ancient tradition, it acts as its final filter. Both paradigms rely on each other. Ancestral hypothesis on the left, scientific proof on the right, overlapping at modern efficacy. The modern pharmacy relies on millennia of wild human beta testing. Modern lab testing simply evolved traditional healers' safety-first mindset. The timeline from discovering a strange purple mushroom to synthesizing a life-saving pill is one unbroken chain of human ingenuity. When evaluating any mind-altering or medicinal plant, respect the ancestral tradition that discovered it, but demand the scientific data that proves it is safe.